All right, well, you're recording. Okay. Thanks for the invite. I'm really happy to be here. This is my lab. These are most of my students here. I'm at, as I mentioned, I'm at in the real London in Ontario, Canada, which is, if you go from Detroit to Toronto, which are maybe cities you guys know, in the middle is London. And then I don't even live in London. I live in the middle of nowhere. So that's why there's nice green space here. So these are the people who are doing the actual work. I don't work in the lab anymore. So any of the work you'll see is from my students. So you always have to remember the people who do the work are not necessarily the people who are here. Okay, so last night I got asked the question from Kat, why do you work on NARS? And I actually have not worked on NARS for very long. I work on NARS because I met Rachel and Terry. So in 2023, I organized a conference together with my husband, Patrick, which is the one big synthetase conference that we have. We have that every other year, and it'll be following this conference here. We'll stay in the same hotel and have this conference. And for the first time at that conference, this conference has been running for about 30 years, and it's all about the different synthetases. And we thought we should invite patients and patient caregivers and patient advocates to connect scientists with the researchers, because us as researchers, we really don't have, I'm not a physician, so I don't have any contact with any patients. So I was like, maybe we should get those people together so that it provides meaning for the patients, and it also provides a lot of meaning for us. So not just having the numbers. So I know all the synthetase numbers, right? The mutation, we know all the numbers, but I never know what's behind that. And if you get what's behind that, it has much more meaning. And I can see it in my students when they realize there's actual people behind them, they light up and they want to do the research much more. So this is something that you as a patient organization, as a foundation, give meaning to our research. So it really is, it made all the difference. And after the synthetase conference, we got emails saying, hey, this was the greatest thing ever. And we're having it at this conference again because it was so successful. So at the conference, when I looked through the abstracts, I saw an abstract from somebody named Dr Heineman, and I was like, I don't know her. But it's like, oh, this is very disease related. Maybe I should ask her to be on this patient panel. And Terry came, and then Rachel said, well, maybe Terry should come and be on the patient panel. And it was really great. And then because of that, I was like, oh, I don't work on NARS, but I could, because there's not too much NARS research going around. So I work on all kinds of different, NARS is one of 20. So there's 20 amino acids, asparagine is one of them, and NARS is one of the synthetases. So there's 19 others. There's actually 37 in humans, because we like to have things in duplicate. So I worked on other ones, but not NARS. But it's like, I have all the resources to do it, I have the knowledge to do it, so we started working on NARS. So from then on, that was in 2023, summer 2023, I hired a new PhD student, Tristan, who's been stellar. He started in January 2024, and most of the work you'll see is from Tristan. I received funding from the Rory Bell Foundation in 2025, and the funding we got from Rory Bell enabled us to do work and then apply for more funding. So then Tristan got funding for his whole PhD. So I will never have to pay Tristan myself again. So the funding we got for Tristan's PhD was $90,000. Canadian dollars, we're not US dollars, so not the same thing. A little less, maybe like 60,000, 70,000 US dollars. So funding from Rory Bell helps us get more funding for more research. So it's sometimes this, we call it seed funding, is really what gets research kickstarted, right? It won't supply my whole research lab, but it does supply enough to do stuff. Okay, so the research goals of my lab is to study why the mutations that your children have cause disease. So your children are all very different, and the mutations that your children have are all very different. So it's not just one NARS disease, the symptoms are different because they are in different parts of this lovely little protein here. You can see I highlighted some of the mutations here. So we work on the protein level, as did Ingrid, and I'm not showing any of what we're doing there because Ingrid beautifully did a lot of that work and just published that. But so we look on 3 different kind of levels. So the first level is we look at the actual protein, so the actual NARS protein. We make it in the test tube and look what does it do. Does it do something different from what it's supposed to do? We also use yeast models. So yeast, as you might think, why do we work? Yeast are not humans, right? No, of course yeast are not humans, but yeast are actually very similar to humans. And yeast are super easy to work with. You can, make beer, make bread with yeast. They grow super fast, and they're really cheap. So in yeast, we can look at all the different mutations very quickly without, taking 20 years to do it. So if you want to make a mouse for every mutation, that's very hard to do. So sometimes going to a simpler organism helps us do things faster. And then the work I'll talk about today is actually in human cells and patient-derived cells, where we've made so far the most progress. We are making progress on yeast, but I won't talk about that today. Okay, so the Rory Bale Foundation supports our work in finding out why NARS mutations cause disease, and then also testing different therapies to see if they will help. And we can do that in both yeast and in human cells. Okay, so now we'll go into the actual science, and I'll try to explain it, but you're welcome to interrupt me. And if I talk too fast, I tend to be a very fast talker. You just let me know if I have to slow down. I'm too fast? Okay. Okay, so this is the NARS protein here. The NARS protein makes a dimer, meaning NARS doesn't like to be alone. It always partners up with another NARS, okay? So when you have a dimer, you have things like how does the dimer stick together? And lots of Ingrid's work has shown that sometimes that's a problem. It gets too sticky, or if it doesn't work right. So here's your NARS protein. So what we did for this work is we put a gene encoding NARS into human cells. So this is not patient cells. These are just regular lab cells, okay? They're healthy lab cells that we can grow fairly quickly and fairly easily. So we put the bad variant, or the disease-causing variant, into regular lab cells. The reason why we do that is in this way we can control what we're doing. We're only changing the mutation. The rest of the cell is the same. So if I were to compare patient cells from your child to me, that's a different person. So there's a lot of different other changes. So for us to prove that something's going wrong based on just one mutation, we have to change just one thing at a time. So that's an important part in science. You change one thing at a time, and then you see what's going on. Okay, and then in the cell, we have the regular copy that's already in the cell, and it can make this nice, really heterodimer to study your disease. And then we see, are the cells alive? Are the cells happy? Do the cells do stupid stuff? What we found, and now we worked on 2 different mutations, so these are those numbers, R522X and R534X, and what those mutations do, they're nonsense mutations. They stop the protein earlier than they're supposed to. So all the work I'm showing is just on these 2 mutations. So it takes time, and I can only make my students do so much work. Apparently there's laws about that, where you can't make them work all night. So they study a few mutations at a time. Okay, so what we look at is it different between when we put in the wild type protein, so wild type for us means the healthy copy, or the mutant copy. And what we see here, I mean, there's little bar graphs, but you can see that this black one is lower than this one. Okay, so and then we do statistical analysis to, is this really different, or is it just by chance? And you can see if we overexpress the mutants, both of them have a higher cytotoxicity than the wild type one, which means the cells grow fine, but they die really quick. So for these 2 mutants, if you overexpress them in human cells, you see that they start dying off. So for us, this was great news because we can confirm this actually has an effect. It's not just, we can statistically say this is actually disease causing. And we have a phenotype. So once you have a phenotype, you can start trying to fix the phenotype. If I don't see anything in the lab, I can't work with anything. Okay, so I need to find something that's different so that I can find out how to fix it. Okay, and then this is some of the some other data we have. They don't just make the cells grow slower or die faster. The NARS mutants also cause something it's called the integrated stress response. So when your cells are stressed, they turn on a stress response pathway, which is great because it's supposed to help the cell deal with the stress. What's not so great is that we also know if that's always turned on, that causes disease in itself. So the cells are unhappy, they die faster, and then they react to it by turning on their stress pathway, which can then further contribute to disease. So this is something that's known for other diseases. We know chronic activation of the stress response is not a great thing. So we don't necessarily want that either, even though it seems like a logical thing. We want to react to stress. So this data basically shows you, again, there's some weirdness going on here, but it shows you that this is turned on. And I know the bar graphs, this really is kind of funny displayed here, but we can see that there's an uptick here in how high the bar graphs are. And when we do statistical analysis, we can see this is actually happening. Okay. Okay, again, this is like the slides are a little funky, but now we go to trying to find ways to fix it. And Ingrid explained that the NARS protein has a job, right? It's supposed to take asparagine and add it to something called tRNA asparagine. So what the protein has to do in the cell, it has to find asparagine and it has to find the tRNA. If you just do this in the test tube, you know, you give it tons of both, it might work or not. But in the cell, there's a million different other things going on. So it really has to be very specific in finding asparagine. And if you have a protein, so we know from Ingrid's work that the 534 stop mutation is really bad at doing its job. And it's like you ask your child to go in a soccer field and find a red ball, and you put 10 red balls in a soccer field. It might take it a while to find the soccer ball. But if you put 100,000 red soccer balls into the field, it'll have an easier time finding it. So this is what we're doing with asparagine supplementation. We just give it a ton of asparagine. And asparagine is an amino acid you can buy in the drugs, any drugstore. It's well, maybe not, okay, some drugstores. Rachel said it's not that easy. But basically, you can buy that, and it's not a regulated drug in itself. Okay, and we just say, okay, we'll put a ton on there and see if that makes a difference. And it did make a difference. So you can't really see it well here because the stuff is a little bit messed up. But in black is our wild type, and in blue, you can see from black to blue, it goes up. And then it goes back down when you look at this green box. So the bars are supposed to be colored, but they're not. So you compare black to blue, it goes up. So this is your mutant doing its job poorly. And then the cytotoxicity goes back down when you add asparagine. So by adding the amino acid to, now remember this is in cells, we're not experimenting on humans, so this may or may not work in humans. But the cytotoxicity goes down. So we're able to rescue the cells, we're able to help the cells grow better by giving them asparagine. And then it has the same effect when we look at as I said, the stress response is on, the stress response goes back off when we add asparagine. So it works on not just the cell growth, it also works on the stress response. We have different approaches. So the other thing that the synthetase needs to find is the tRNA. It has the two substrates. So we did the other thing. We just added a ton of tRNA. And tRNA does the same. It helps rescue the cytotoxic defect, so it helps the cells survive better. But it does not prevent the activation of the stress response. So tRNA works to some extent, but not quite as well. Okay, and then finally, we tried just to turn the stress response off because we know that's not good. And we can do that with a small drug that's usually being used for cancer. So this is part of your drug repurposing scheme where you try to use a drug that's used for something else. And we can turn the integrated stress response off on itself, and this also helps the cells to grow better. So we're trying different avenues to combat the effect we see in cells. Okay, we also looked at then the proteome. The proteome is something where you look at the entire cell and look what changes in the entire cell. And if you just look at the color coding here, the wild type is here, and you can see your mutant has some red changes here. And when we quantify the changes from wild type to mutant, we have about 38 changes. And when we add asparagine, those changes go down. So asparagine is for now our best bet for those 2 mutants because asparagine restores the viability of the cells, so the cells are happy, the stress response is off, and the changes in the proteome are mostly reversed. The 2 other avenues we have, tRNA and the integrated stress response inhibitor, they don't work quite as well. Okay, finally, when I talked to Rachel about asparagine supplementation as a possible way to look at, a treatment for some of the mutations, she said, well, but asparagine, she's a pharmacist, asparagine is converted to aspartate glutamate, and glutamate is known to cause seizures. So do you really want to give your kid asparagine? I don't know the answer, but we did put asparagine on cells and then looked at, do they convert it more to glutamate? The short answer is no. We don't see any of that in cells. So we give the cells a ton of asparagine. You can see it here. There's untreated cells and treated cells. You can see how it's going up for both the wild type and the mutant ones. You have lots of asparagine in there because we give them asparagine. But you can see there's no difference between an aspartate, glutamate, or glutamine levels. So at least in cells, this does not make any, there's no concern from the cellular side. There's probably other, I'm too fast. Okay.
You're getting too excited about
I know. Sorry. Okay. Finally, and this is my last sciencey slide, we looked at patient cells because we've shown this in our human cells, but does this work on patient cells? So we got patient cells from R534 stop codons. I don't know which of the kids it was. Rachel just left, so she would know. Terry knows?
I mean, yes, these are our kids' cells that you worked on.
So we did get the cells from the Rory Bell Foundation. So these are skin fibroblasts. So it's a skin sample. And we grow them, and then we compare them to a control, age-matched control. So if this is a 10-year-old kid, 10-year-old male, we would use 10-year-old male cells. If it's a 5-year-old female, we would use 5-year-old female control cells. And we do see those cells grow slower than the control cells, which in itself is not, sometimes it's just variation from person to person. But when we add a lot of asparagine to these cells, they do grow better, and they grow to a level that's not distinguishable from the control cells. So it works for patient-derived fibroblasts for this one patient. So I just want to caution that I don't want to overgeneralize our findings because we only looked at 2 mutation and we only have patient samples from 1 patient. So we just have to be careful that we're not like one size fits all. Okay, so, and there's some other data here that shows that there's actually there should be bar graphs here. There's very little asparagine. Like if you add asparagine to the cells, they're actually happier also in performing their function. Okay, so this is my little summary slide here. When we have cells, our lab cells or the patient cells, we know that asparagine supplementation reverts the proteomic changes, and it also takes away the cytotoxicity, and it also takes away the stress response. We know that small molecule inhibition takes away the stress response and the cytotoxicity, but not the proteome. And tRNA supplementation really only takes away the cytotoxicity. So different treatment approaches have different efficacy. So some are better, some are not as good. Why are we still trying the other ones? Why are we still trying tRNA and integrated stress response? I know from my other research in histidyl tRNA synthetase that for some patients, histidine supplementation in that case does not work. We have tried this on actual patients. We have a clinical trial for that. So we know for some it really works beautifully, and for some it doesn't work. So for those, we might have to consider some of the other therapies, or we might have to consider combined therapies. So I think this is a case-to-case scenario where we really have to look at every mutation on its own before we can make blanket statements, this is going to work. So because the disease is so diverse, I think we have to just be really careful about saying this is the cure. It might work for some, it might not work for everyone. So I just want to caution you guys. So what are we going to do in the future? We'll work with yeast more to do more drug repurposing. We'll try to use more patient-derived cells to study some other variants that we haven't studied yet. My student, I still have him for another 3 years, so he can do a lot of other work. And we work with collaborators and clinicians to try to bring asparagine supplementation to the clinic. That's mainly, I'm not a clinician, I'm not a real doctor. Yeah, see, I work in a research lab, so that's out of my expertise area. And we will do more drug repurposing to screen for more drugs that we can use to help with symptoms. And then I want to thank my lab. Sometimes we do have fun and go to laser tag or something like that. So my lab's been great. Tristan has been doing excellent work. And then I have a postdoc, Jenica, who just applied for a fellowship based on NARS, and hopefully she'll get that. And everybody for funding, especially the Rory Bell Foundation, which should have been in purple or green. I mean, really, yeah. And that's it. Yes.
Does anyone
All
have
right.
questions? No questions. Explained it perfectly.
Or I was too fast.
Yeah, it's picking it up. I could see it's coming through.
Even out there, Spaz's wife is sitting out there, and so.
Oh, it's working out.
It's working outside.
Yeah. Yeah. And I know later, Ingrid, we're going to talk more about asparagine trials, things like that as we think about drugs, helpful supplements for the kids too. So thank you. Okay. Okay.